Okay, we're rolling. Okay. Hello everyone. We are the Girly Fight-Ins. It is me, Danya, Gabriella, Natalia, and Champagne. And today we're going to be talking about a paper titled Responses to Periphyton Communities to Abrupt Changes in Water Temperature and Velocity and the Relevance of Morphology, a Mesocosm Approach, which was published in 2021. So I'm gonna introduce the paper and set the stage, and then I will pass it to the other girlies. So, um, kind of just as an introduction to set the stage, they were talking about how perifying communities are essential in stream ecosystems, and they play a big role in nutrient cycling and providing food for other organisms. But hydroelectric power plants disrupt the natural flow in the streams, which leads to daily temperature and velocity fluctuations called hydropeaking or HP and thermohydropeaking or THP. And thermohydropeaking, THP, combines flow pulses and temperature shifts which acts as multiple stressors for rivers. And understanding how THP or thermohydropeaking affects the structure and function of periphyton communities helps inform conservation strategies for rivers impacted by hydropower. So the objectives for this paper was to assess those multiple stressor combined effects of THP on the structural, which is chlorophyll A, and pigment composition, and the functional, which is R, ETR max, the glucosidase, and phosphatase activity. Um, so both of those structural and functional characteristics of the periphytic community in that river. And so now I'm going to pass it on to Gabriella with the methods.
Thank you, Dania. The experimental design was set up in Lower Austria at their high-tech facility. This was very conveniently located downstream from Lake Lunds. So in that facility, they established six flumes of 20 meters long, and they divided them into two pool and two riffle sections each. They developed these sections utilizing stones as well as clean sediment to best assimilate or look like what you would see in the lake and in nearby streams. So they separated into two experimental phases, the colonization and the treatment. The colonization focused on observing and measuring the growing of periphyton, whilst the treatment phases, they treated three of those flumes with warm THP and three with cold THP. They did this by utilizing water from the epilitmian and the metalitmian of Lake Lunds. And there were different variations of about 5 degrees between each for cold and for warm. In terms of a control variable, so these flumes were not utilized all at the same time, and they were not exposed to the warm or cold THPs all at once. So they had these 12 metal plates with 20 stones that they rotated between these understood flumes, and they maintained these metal plates and stones in their respective pole and riffle sections in which they had been placed originally. Their observations were every 0, 10, 17, and 24 days. And part of the sampling was done by scrubbing 3 stones from each of the sections during each of those interval days. In terms of measurements, they scrubbed the stones for chlorophyll A, as well as utilizing a high performance liquid chromatography or HPCLC analysis. And they also looked at extracellular activity in the shape of A phosphatase, alpha phosphatase, and beta glucosidase. These two, because they were the most closest and possible way of evaluating the degradation of different matter, or organic matter. They also looked at water chemistry. They sampled each flume for phosphate, ammonia nitrate, and silicate to account for diatoms. They also evaluated or observed water temperature by using data loggers before and after each pH treatment. And they also looked during the THP treatments by utilizing temperature probes. They also looked at flow velocity utilizing a current measuring device during the THPs and across all of the sections, so both the riffle and the pools. Um, and once they scrubbed those stones, they looked at the biofilm slurry that it formed, and they used it to look for the paraphyletic chlorophyll A and the pigments that were later analyzed with a spectrophotometer. These pigments were detected utilizing methods known from Woidke et al. 1994. They also looked at photosynthetic activity utilizing interferometer as well as a software that produced a rapid light curve as well as single yield determinations for each of the sampling days. And they also used fiber optic device for in situ measurements on the stones. They also looked at enzyme activities. They utilized a sample of that same biofilm slurry and used a 96 well microplate spectral scanning multimode reader. And with that, they were able to measure alpha phosphatase and beta glucosidase. And then they took all of that data, and if you can scroll down a little bit more for the statistical analysis, they looked at normality. First of all, they assessed normality of the data utilizing Kolmogorov and Smirnov tests and F tests. If some of this data did not meet or satisfy the normality assumptions, it was transformed using Box-Cox transformation and again tested for normality. They utilized a Mann-Whitney-U test to detect differences between THP treatments and control for each of the sampling days. And they also used a two-way ANOVA, factorial ANOVA to detect influences of THP on habitat, as well as those two variables interactions. They also looked at most abundant pigments and the parameters for biomass. They used those adapted principal response curves, so utilizing again the curves that I mentioned before, to evaluate the pigment development during and between treatments. And they also use multiple regression analysis for key abiotic, such as temperature, treatment, habitat, index of treatment, and biotics, such as percentage of concentrations, factors for alpha phosphatase and beta glucosidase. Now I'm going to pass it to Natalia for results.
Yes, so the authors concluded that the thermohydropeaking had a structural functional effect on periphyton communities. And they made an important differentiation between different changes, either if it was a cold or warm temperature, and if it was a pool or a ripple treatment. So specifically for chlorophyll alpha, in the pool sections, they found that chlorophyll alpha showed a significant increase at cold temperatures. However, they did not show the same pattern in warm treatments. Then related to the other pigments they measured, they made an emphasis of chlorophyll alpha and fucoxanthin because those two were the ones that were at high concentration during the whole experiment. And they showed that those two pigments were influenced by treatment and habitat. Treatment referring to cold and warm temperatures. Then regarding our photosynthetic activities, they showed that there was not an effect by treatment, but they did show an effect by habitat. And for the enzymatic activities, they showed that alpha phosphatase activity showed a significant treatment effect, but not habitat or the interaction. And for the beta glucosidase, activity, it was not affected by treatment, but it was affected by habitat. And there was also an interaction between treatment and habitat. So those are the main results the author showed.
Okay, and time for discussion. So even though there was some data that was collected during the summer, this algal measurement was comparable to the measurements that were collected in the winter alpine streams. But it's important to remember that the species collected could be different in different seasons. Our researchers confirmed their first hypothesis and found that the influence of THP on algal biomass depends on the direction of temperature change, whether it's cold or warm, and habitat type, whether it's pool or riffle. Our flow velocities can affect the morphology by increasing shear stress. Thermohydropeaking can also interfere in functional processes of the periphytic community and has a tendency to promote photosynthetic activity. And then our photosynthetic activity increase might be more pronounced during our winter months. Our warm THP is independent of the morphological settings. However, this pattern was more pronounced in our riffle section, where higher flow velocities could favor algal nutrient uptake. Our conclusion, our periphytic algae function as ecosystem engineers, and they're also strong algal-bacterial coupling in the THP impacted streams. The coupling can lead to severe effects on nutrient and carbon cycling of a river, and it highlights the importance for mitigation measures in these rivers and streams. In our winter months, the algal biomass is especially depressed in riffle sections, and researchers observed more pronounced impacts in the cold thermohydropeaking than in the warm ones, due to a stimulation of diatom growth in these colder hydropeaking areas. The effects of warm thermohydropeaking will be more pronounced in winter months when cold water temperature adapted species dominate the periphytic community. And the results showed, our results showed clearly that these riffle sections would develop less periphytic algal biomass and activity. So our photosynthesis and extracellular enzyme activity are what we're referencing. And then thermopeaking can therefore promote the development of a large areas with less biomass availability for primary consumers. And that's it from the Girly Phytons.
Thanks for watching. And